Welcome everybody to our today's webinar with the title Supervised Learning of Disease-Associated Cell States and Dynamics. And my name is Dennis Gülshoff. I am a co-founder of Scailyte. I am now working in Scailyte's machine room, like developing and improving our core algorithms. And at Scailyte, we're using these algorithms to gain clinically relevant insights from high-dimensional single-cell data, such as single-cell RNA or cyto data. And we're collaborating with various companies, particularly in the areas of cell and gene therapy, immunology, or oncology therapies. Our core technology of our service is our SkyVision platform, which allows us to efficiently discover endpoint-specific expression patterns or cell populations. And interestingly, this core technology, SkyVision, was originally based on CellCnn, an algorithm published in 2017. And therefore, it's a special pleasure for me to introduce the PI contributing to that paper, my longtime friend and co-founder and now chief scientific advisor of Scailyte, Professor Manfred Claassen. So Manfred has an interesting yo-yo movement-shaped CV, where he started graduating in biochemistry and computer science at the University of Tübingen, then went on for a PhD at ETH Zurich. Then he came to the most distant point from his roots and joined Daphne Koller's artificial intelligence at Stanford, at the intelligence lab at Stanford for a 2-year postdoc. And from then, he went back to ETH, but this time as an assistant professor for computational biology, and then finally back to his roots in Tübingen, where he became a professor for clinical bioinformatics at the medical faculty 2 years ago. So Manfred's research focuses on learning explainable quantitative models in systems medicine from high-dimensional single-cell omics data. His group develops representation learning algorithms to identify disease-associated and potentially rare cell populations, very similar to what CellCnn does and also our SkyVision platform. And the computational models that his group develops can ultimately enable comparative single-cell studies, for example, in predicting immunotherapy response in melanoma or the diagnosis of multiple sclerosis. So all this is very similar to what Scailyte does, and it's no coincidence that Manfred was a co-founder of Scailyte. On a technical note, we will now be listening to Manfred's presentation. After that, this will be followed by a QA session. And I would ask you to mute yourself during the presentation and use the chat box on the bottom right side of your screen to ask questions already now. So now I'm looking forward to the presentation and the discussion afterwards. And with that, I hand over to Manfred, and the stage is all yours, Manfred.
All right. Yeah, thanks a lot, Dennis, for the nice introduction and being reminded of all the good old times. Yeah, and I'm very happy to give you an update on some of our research vignettes that I try to subsume in this title, Weekly Supervised Learning of Disease-Associated Cell States and Dynamics. I will just jump in right away, like at a very high level, what I am doing or what we are doing here with me having my research hat on. So we are very interested in high-dimensional single-cell data that is depicted here in this cartoon. So here have the cartoon of a tumor that is composed of a multitude of cells, different types of cells, partly being tumor cells, and then cells corresponding to blood vessels, and then cells corresponding to infiltrating immune cells, to name a few. And what you're probably all aware of is that nowadays it has become a lot more easy to measure high-dimensional molecular profiles of these cells. So at Scailyte, we started to be very excited about mass cytometry, a single-cell proteomic technology. But besides mass cytometry, there are also quite a few different other fluorescence-based flow cytometry techniques that are now equally potent in order to get these single-cell proteomic profiles. Besides this, obviously, there have been tremendous advances in single-cell sequencing, allowing to today pretty routinely measure single-cell transcriptomes, and now also increasingly other single-cell omics modalities, as for instance, single-cell epigenetic profiles and so on. And besides this, there also has been tremendous developments in the development of imaging techniques that allow to measure many molecules at the same time at the single-cell level in the spatial context. There, for instance, we have imaging mass cytometry, codex, maxima, and so on. And all of this is obviously very exciting. The data that comes out of these technologies is very information rich, but oftentimes this data by itself doesn't tell you the answers to the questions that you have. And so, for instance, questions that you might have in translational projects is, how can I see from the single-cell profiles whether a patient has a good or a bad prognosis? And what we develop in the lab, in our lab here, are machine learning methods that allow us to go from, I don't know, can you see my pointer? I guess now, can you see it?
I don't see it right now.
You don't see it? Let me see. I think I can. Yeah. So now you can see something, yeah. Right? Yeah. Okay, great. Where were I? Okay, so we develop these machine learning methods that allow us to go from the left-hand side, from these high-dimensional molecular, single-cell molecular profiles to, generally speaking, macroscopic phenotypes like disease states, therapy response, patient outcome, these kinds of things. And more generally, the type of machine learning tasks that we address and for which we develop methodology in our lab are, on the one hand, so-called comparative single-cell data analyses, where we assume a situation where single-cell data is acquired for patient cohorts that have different macroscopic phenotypes, and we want to compare, like disease, non-disease, and figure out what are the patterns in the single-cell data that distinguish these conditions. A second branch of analyses, very similar, are these comparative spatial single-cell analyses, where we assume a similar study type, but instead of having suspension-based single-cell data, we assume that we have single-cell data that has spatial resolution, that looks like an image of a tissue overlaid with all this molecular information. And then the third branch of techniques that we look into are comparative single-cell time series analyses, where we in particular focus on single-cell RNA sequencing data and try to understand how different cell types are transformed into each other by signaling or differentiation processes. All right, let's jump into the first vignette that is related to comparative single-cell data analysis or comparative spatial single-cell data analysis. So here I show you a diagram of the CellCnn model that at least a fraction in the audience, I guess, is very familiar with. So this is from the good old days, this simple convolutional neural network model that uses single-cell data input here on the left-hand side, this data matrix, and that uses this simple convolutional neural network in order to associate this input with phenotypes. And one question that we asked at some point still at my time in Zurich was whether we can do a similar analysis where we do not consider mass cytometry or flow cytometry data, but imaging flow cytometry data. And so this was a collaboration with the De Mello Lab at ETH Zurich. And what the De Mello Lab is known for is for the development of all kinds of sophisticated methodology to measure single cells. And one of the methodologies that the De Mello Lab develops is imaging flow cytometry. And here you see an overview of this imaging flow cytometry machine that the De Mello Lab built. in a nutshell, what imaging flow cytometry does is to come up with a microfluidic setup that allows the imaging, the bright field imaging of single cells. And the device that the Demello lab has built is a device that allows to image individual cells at 10x magnification. And the types of images that you get, you see here on the right-hand side for individual images. And the quality of the images is obviously not like from a still bright field microscope, because the cells flow past the microscope very quickly. So you only have very small exposure times in order to capture or to get an image of a cell. And for this reason, the Demello lab has developed all kinds of techniques in order to still illuminate the cells sufficiently quickly and strongly in order to get a bright field image in this very small amount of time that the cell floats around the microscope. And the beauty of this method is that in a high throughput mode, you could in principle image 100,000 cells per second. For the study that we considered, we actually considered a lower throughput setup that allowed us for a whole study to measure in the order of 10 to 100,000 cells. And now the question is, can we identify cell subsets based on their morphology alone that are disease associated? And considering the following setup here. So here we consider a setup where, again, we have a patient cohort for which we extract, in this case, PBMCs, peripheral blood mononuclear cells, that are then analyzed with imaging flow cytometry. So for every single patient, you get this collection of single cell images of their PBMCs. And now what we know about the patients is their health status, whether they are healthy or diseased. And now what we would like to do is to classify, on the basis of the imaging flow cytometry data, the disease state of the patients. And this is something that has been tried in the past, to use imaging flow cytometry in conjunction with machine learning approaches in order to do this. And the typical approach is depicted up here. So where you went ahead and in a first go, you had a human expert, a clinician, going through tens of thousands of these images and annotating the cell images as cells that come from a healthy donor or from a diseased donor, or to classify cells that have a disease phenotype, to be more precise. Because it might well be the case that not all the cells in the patient blood are cells that are associated with the disease, because there are just quite many cells floating around in a patient's blood, even in a diseased patient that are not don't have a disease phenotype. And now, after having gone through this annotation process, you can now go ahead and train a conventional computer vision classifier that uses this so-called strong supervision for the image at the single cell level, whether this is an image of a cell that is diseased or comes from a diseased patient or from a healthy patient. And this is an approach that works pretty well as long as you have sufficiently many labeled examples. And there you can already see where the problem arises. So the labeling of the data is obviously a costly process and is certainly a bottleneck for these types of approaches. And now the question was, can we go ahead and circumvent this manual annotation of the images and just come up with a weakly supervised machine learning model that uses a collection of single cell images that might comprise a fraction of disease phenotype cells, and then use this collection of images to make the classification. And this is exactly the same type of problem that the CellCnn model solves for single cell data, where for every single cell you don't have an image, but you actually have a molecular profile as information, as feature vector. All right, and so basically, I'm glossing over some detail here, plugging this CellCnn idea into this setting here. Maybe just as some technical background here, in order to make the image data look more like some sort of flow cytometry data that is in the order of 30-dimensional, what we did is that we used autoencoders to learn an unsupervised representation of these images in order to get as input for the CellCnn model a collection of representations of these single-cell images for classification. And the type of application that we considered for this CellCnn variation was a collaboration with Emanuela Genova that probably also a lot of people in the audience know quite well, because Scailyte also worked closely with her in a related project. And so what we wanted to do here is to diagnose Sézary syndrome, but not using molecular information, but only on the morphology alone. And the reason why we chose this application is because the cells that are related to the Sézary syndrome that manifest themselves in the blood have a funny morphology. So they have a so-called cerebriform nucleus. So that is, they have a nucleus that has a shape, a form that basically looks like a small brain. So, and this morphology we hoped is something that such a model could learn in a data-driven fashion, even without annotation, as I described before. On this slide here, you see how well we did. The cohort of patients for which we had samples that we could analyze using the imaging flow cytometer was actually quite small. So we had 5 patients, 5 Sézary syndrome patients, and we only had 4 healthy donors that we could analyze in this context here. And so here on the right-hand side, you see the performance of the model that we propose on the right-hand side, and then on the left-hand side, the performance of the model that basically trains a classifier on single-cell images, where the single-cell images are just labeled all according to the disease phenotype of the patient. So possibly also labeling a few or quite many cells actually that do not have a disease phenotype as looking as a diseased phenotype cell. And then in the middle, we also have this conventional approach that is referred to here as expert labels, where we actually had experts sifting through all the single-cell images that the imaging flow cytometer generated, and then annotating these cells according to their disease phenotype in an expert fashion. And what you can see here is that the separation of the two classes is actually the best when using these weak labels, using these CellCnn variation analysis in order to classify the patients. And so with this, I'm already at the end of the first vignette, and I would like to move on and talk about a second vignette that is the analysis of spatially resolved single-cell data. And in order to motivate this, I maybe just take a brief anecdotal detour of a study that we did before all this high-dimensional spatial single-cell biology techniques took off as they are taking off right now. And that is a computational pathology project that we performed together with pathologists at the University Hospital in Zurich with Peter Wild, who is now in Frankfurt. And so the reason why we started to work on this is because we thought, okay, so this histology data that pathologists look at is basically single-cell or spatial single-cell data. It is just not very high-dimensional. In this case, where you have this H&E staining that basically highlights morphological features of cells. And so the basic idea was to go ahead and reconstruct or annotate these histology images with respect to the severity of prostate cancer, and doing so by a deep transfer learning approach. And so what you can see here on this slide is an overview of the different morphologies of prostate tissue in prostate cancers of different severity. And so in the upper rectangle, you see the arrangement of glands in the prostate in the healthy tissue, and you basically have this very uniform arrangement of the glands in the healthy tissue. And then with the increasing progression of prostate cancer, this uniform structure is more and more disrupted and becomes more and more chaotic. And so what we thought in the beginning is that we basically go ahead and take images that were annotated by pathology experts and then use a deep convolutional neural network to reconstruct the labels annotated by the experts. And for this, we used a deep convolutional neural network. This is how the annotation of the pathologist look like. So here you have these tissue microarray slices of tissue with different examples of different Gleason grades of different prostate cancer severities as segmented by the pathology experts. And this was the input data that we used to train this deep convolutional neural network. And in the end, this is the performance that we achieved. So here you see the confusion matrix of the annotation of the pathologist on the y-axis, and on the x-axis you have the corresponding annotation of the model, of the trained model. And what you would like to see on this confusion matrix is coincidence of the model annotation and the pathologist annotation. That is, you want to see dark blue color or large numbers of coincidences on the diagonal. And if you look at this, you see that you have a concentration of coincidences around the diagonal for the first pathologist and the second pathologist. You might ask yourself, well, is this now a good performance or is this a bad performance? And the reason why this is a little bit difficult to assess is that the annotation of the Gleason grade in itself is a subjective measure. And in order to see how well we do, we compared the two different pathologists and evaluated the coincidence of the annotation of the two pathologists. And what we saw was that the degree of coincidence between the two pathologists that were so kind to annotate the data for us was exactly at the same level as the discrepancy between every single pathologist with the model predictions. Something very nice that was very important to us is the possibility to interpret the trained model with respect to the types of patterns that the model focuses on in order to make a decision. And so here I'm showing you an example of using class activation maps to do this interpretation of the deep convolutional neural network. On the left-hand side, you see an image patch of prostate tissue. So here you see this glandular structure. And the model correctly annotates this image patch as an image patch corresponding to Gleason 3 severity. And you have highlighted in these reddish, yellowish colors, red being like more important, those regions in the image that are particularly important for the model to make this decision that this is a Gleason 3 pattern. And when we showed this to our pathology collaborators, they were very excited because they said, this is exactly where we look at in order to make the decision whether this is a Gleason 3 or the very similar morphological pattern Gleason 4. And that is because these reddish areas exactly map between in these regions between the glands. And the difference between Gleason 3 and Gleason 4 is the increased frequency of fusion events between the glands that disrupt this uniform arrangement of the glands. So that was quite nice. And this is something that I'm going to revisit in the next application that is actually closer to our heart. And that is doing high-dimensional molecular imaging of tissues. And so, as you probably know, there are quite a few upcoming in situ proteomic imaging approaches that allow you to measure dozens, actually up to 100+ different protein markers in the same tissue slide and image their location with single cell or even subcellular resolution. And these techniques are, for instance, Codex or Maxima. And what we headed out to was, in collaboration with partners at the NIH, Tim Greten and Benjamin Ruf, was to understand how the cell type composition in liver cancer is different around different types of immune cells in healthy liver tissue and tumor liver tissue. And just to give you a little bit of context, the type of immune cell whose surrounding we wanted to characterize are mucosal associated invariant T cells, MATES. So this is a type of innate-like T cell that has been found empirically to be associated with survival of HCC, of liver cancer. And more specifically, if the tumor is infiltrated with fewer MATE cells, then the survival of patients seems to be a lot better than for those patients that feature many of these MATEs in the tumor cell. And what we wanted to know is what is the surrounding of the MATEs, or what is the characteristic surrounding of the MATEs in the tumor compared to the healthy tissue. And the type of experiments that Benny Ruf did was to measure liver tissue comprising healthy tissue and tumor tissue using CODEX, so one of these high-dimensional in situ proteomic imaging techniques. And so here on the left-hand side, you see an image of cells distributed in liver tissue, and you see highlighted the fluorescence signal with respect to 3 different markers that are related to the identification of these MATEs. And so I only show an image that has 3 markers here, but in fact, the number of markers that we considered in this study was 38 in total. And now you can go ahead and look at the data, and you can look at the data as you look at conventional suspension-based single-cell data using dimensionality reduction in order to figure out what kind of cell types you have in there. I will not go into too much detail here. Maybe just as a side remark, the UMAPs or the projections that you produce from this imaging data are actually a lot more convolved and not so clear as you are used to see from suspension-based techniques like single-cell RNA sequencing or mass cytometry. And now, again, in order to identify the typical surrounding of a MATE cell in these different types of tissue, so the tumor tissue and the healthy tissue, you again face a classification task. And since the type of data that we are looking at is an image, one could just think, okay, maybe I just throw a convolutional neural network on it using image patches of images of the surrounding of MATEs in order to figure out what is the difference between image patches in healthy tissue and tumor tissue. But now it turns out that we could do better, at least conceptually, thinking about it beforehand. And that is by also considering the type of image that we are working with. And so the type of image that we are working with is an image of a tissue. And we know that tissues have a specific architecture. And this architecture revolves around the atomic unit, namely the cell. So we thought that we can do better if we take into account this information and use cell segmentation algorithms in order to take the image and segment individual cells and use this information for classification of the MATE surrounding that is characteristic in healthy or tumor tissue. And so the type of analysis that you can think about is to consider a cell segmentation of the image, and then using the cell segmentation of the image to produce molecular profiles of every single cell as for suspension-based data. And then on top of this, obviously also using the location, the XY coordinates on the image of every single cell for the analysis. And as you will see in a second, you can then reduce this analysis, this classification to a CellCnn analysis. And in principle, you could also go ahead and use the cell segmentation and not operate on collections of cell profiles, but you could also operate on collections of cell images and use the approach that I just showed you before for classification using imaging flow cytometry data. But I'm not going to talk about this today. So how can we reduce this analysis to a CellCnn analysis? And so here on this cartoon, you see the basic idea. On the left-hand side, you see a cartoon of the tissue image. And this tissue image comprises regions of normal tissue in green, red corresponding to tumor tissue. And then this is just well, maybe a more biological context detail. You can also distinguish a so-called rim region that surrounds the tumor and separates the tumor, the core tumor tissue from the normal tissue. And now what we can do in order to understand how the surroundings of the mate cells, as depicted as blue dots in this cartoon, how the surroundings of these mate cells, the cell type composition of these surroundings is characteristic in the normal and in the tumor tissue, and for that matter also in the rim areas. You can basically use the in situ proteomic imaging data and the cell segmentation in order to first identify all the mate cells in the image, and then extract k-nearest neighborhoods, and from these k-nearest neighborhood define multi-cell inputs, collections of cells with their molecular profile, and use these multi-cell inputs as input for a conventional CellCnn model and classify with respect to the location of the multi-cell input being either normal, rim, or tumor. And so here you see one of the results of applying this model in order to classify the cell type composition around mates in the tumor and in the normal tissue. And in this example here, you see that the model identifies a group of cells that around mates is more frequent in the normal liver and in the rim, and is less frequent in the core tissue. And now you can go ahead and then do the interpretation of the model. So in a similar fashion as I showed you for this computational pathology project, investigating what kind of morphological patterns are important to make a decision. Here now you can ask what kind of cells are important to make a decision. And without going into too much detail here, one group of cells that is identified by this model is a specific subset of macrophages that is enriched around these mate cells in the tumor. And you can also go back and map the cells that the model identifies to the tissue images. So here on this slide, you see an abstraction of the images taken for a collection of patients. And in pink and green, or different green tones, you see the annotation of normal, rim, and core tissue sections. And then you see as these circular holes, you see the neighborhoods around the mates that were considered as input for the CellCnn model. This is the reason why we internally call these plots cheese plots, because they look like Swiss cheese. And so you also see then in dark red the distribution of the cells that are selected by the algorithm. And you can see quite nicely that the selected cells seem to be much more frequent in the normal, and for that matter also in the rim region compared to the core tissue. And so here you see another summarization of the result. So you can look at the selected cells and analyze what kind of cell types these cells are. You can also look at the molecular profiles of these cells. And so here you see the frequency of the selected cells in the normal and in the tumor tissue. And so this is one cell type here that I mentioned before, these macrophages, these M2 macrophages, that are PD-L1 positive, that are identified by the model. And our collaborators also performed validation experiments in mice in order to investigate the role of these macrophages, and performed experiments where they deleted these macrophages, and then figuring out that the cells that, or the mice that are depleted of these macrophages produce mates that are impossible or that do not infiltrate the tumor as strongly as before. All right, and now I'm wondering how much time do I have left? I think it's about 45 minutes or so that I talked.
Yeah, normally, well, I think you're around half an hour, so I think it's okay. Since up to now I don't see any questions. I have a lot of questions, but I can hold back. And how much longer do you think it would take you?
I mean, I guess I can go through the rest in 10 minutes. I hope I do it quickly. So this is, I don't know how interesting this is for Scailyte itself. We personally, find this very interesting. This about like this time series analysis. But let's see. I will go through this quickly.
Yeah, I would love to see it. I mean, to everyone, if you have questions, please use the chat box on the bottom right to ask questions already now. And yeah.
All right. So this vignette is about reconstruction of differentiation trajectories in the context of chronic infections, viral infections, and specifically considering differentiation of T cells. And so what you can see here is a very simple cartoon of what happens if you get an acute virus infection from a T cell perspective. So you typically start off with naive T cells, and here we focus on CD8 T cells. You start off with these naive CD8 T cells, and some of these recognize virus antigen and are stimulated and triggered by this exposure, and then differentiate into so-called effector T cells that then help the host to eliminate cells that harbor the virus. And then some of these effector T cells, or at least some of the CD8 T cells, to be more precise, further differentiate into so-called memory T cells that then reside for a long time in your body, and then allow the immune system to respond rapidly to a second virus exposure. And now it turns out that some infections have a different development, that is chronic infections, where you have the exposure to the antigen, development of naive CD8 T cells into effector T cells, but then a slowed down clearance of the virus and development of a so-called exhaustion phenotype, that is T cells that seem to be impaired in their functionality. I mean, to produce cytokines to kill virus-infected cells. And what we wanted to understand in this project is how is the cell state sequence that naive CD8 T cells undergo in acute infections, how this is different to the cell state sequence in chronic infections. And so since this is a process that evolves over time, that evolves over quote unquote long periods of time, so the type of model system that we investigate is lymphocytic choriomeningitis virus infection in the mouse. So there we talk about about time spans of about 3 weeks or so. So in order to account for this time span, we have to think about experiments where we measure multiple times over time. And then the process that we talk about is a process that does not involve a single gene or a single protein, but it involves actually quite many genes. So we have a multifactorial problem or process. And also we have a process that is asynchronous. That is, so when the host is infected with a virus, it's not the case that every single naive T cell sees the virus at exactly the same time and undergoes the process with the same timing. And in order to be able to describe, to measure, to monitor this process appropriately, we thought about doing a single-cell RNA sequencing time series experiment, as depicted here. So we have mice that are exposed to virus, and then over the course of 3 weeks at 8 different time points, we extracted T cells from the spleen and performed single-cell RNA sequencing. And so specifically what we did here is to use adoptively transferred P14 cells, which are transgenic CD8 T cells that have a transgenic T cell receptor. So allowing us to investigate a pool of CD8 T cells that is very homogeneous with respect to their capacity to respond to virus exposure. And so here you see a T-SNP projection of the single-cell RNA sequencing data coming out of the acute infection experiment. What you can see by looking at hallmark genes is that up here you have naive T cells, here you have these effector T cells, and down here you have memory T cells. Now you can ask the question, how do the cells get from the naive state to the effector and to the memory state? And you can ask the question whether it is a linear process where you have naive cells developing into effector cells and then some effector cells developing into memory, or whether there is some cell fate decision beforehand, before the stimulated cells develop into effector cells that make the cells sometimes decide to become memory cells. And so what we did here is to perform trajectory inference to answer this question using RNA velocity. And for those that have not heard about RNA velocity, just as a quick primer, the idea of RNA velocity is to use the single-cell RNA sequencing data not only to get an expression profile for every single cell, but also get a direction in which the cell is moving in expression space from that data. And the key idea of being able to do this is related to the fact that single-cell RNA sequencing allows you to measure fully spliced mRNA and partially spliced mRNA. And the profile of partially spliced mRNA for every single cell, you can use to estimate the near future state of the cell. And this is in particular interesting when the cell is changing its transcriptional program, then there is a discrepancy between the unspliced and the spliced profile, giving you the directionality and the speed with which the cell moves through expression space. And so basically what you end up with using this RNA velocity analysis, there's a lot more to it in order to get there, but this is the rough idea. What you end up with are these velocity fields that where for every single cell you also get these directionalities. And what you can see for this acute infection single-cell RNA sequencing data set is that the state flow seems to indicate that there is actually a bifurcation of these T cells, indicating that there is a fate decision of these T cells to become either effector or memory cells. And you can do the same thing for the data that we get from the chronic infection. So in here you have an overlay of the chronic and the acute infection data that indicates that there is an additional branch of T cells that has this exhaustion phenotype. And you can also see here that there seems to be a decision region in this velocity field that indicates that these cells make a decision to either become exhausted or memory-like in this chronic infection setting. And in order to analyze this a little bit more systematically, we developed a tool, CytoPath, that tries to infer these trajectories in a simulation-based fashion. And so the whole idea is that you get these velocity fields and you can then define a Markov model with the measured cell states corresponding to the states of the Markov model, and these velocities allowing you to define a transition matrix, a probability transition matrix of the Markov model that describes the directed probability of transitioning from one cell to another. And then you can use this Markov model to perform simulations, as you can see here, of cells in this region of naive CD8 T cells, and then basically from these simulations perform summary statistics to get these so-called or like quote-unquote average paths from the naive CD8 T cell region to this memory-like region or this exhausted region. And then you can analyze further along these trajectories which of the genes show differential expression over time And again, glossing over a lot of detail here, you see amongst other genes also these known exhaustion or memory genes being differentially expressed over time as you would expect it. And then you can also do fate predictions. You can go back and ask, at what time can I predict with high fidelity whether a cell is going to become memory-like or whether it's going to become exhausted. So what you can see here are UMAP projections only of the region of the naive CD8 T cells with this exhaustion score derived from these trajectories overlaid. And so here on the left-hand side, for instance, you see exhaustion fate. And so you see that the cells that are in this area down here have high exhaustion fate in contrast to those cells in this area up here that have low exhaustion fate. And then you can go ahead and look which of the genes are differentially expressed in these areas in order then to come up with a validation experiment where you try to sort out physically from an infected mouse at an early time point those cells that we would predict to have an exhaustion or a memory fate, and then transfer these cells into an infection-matched host and then assess what kind of phenotype these cells develop. And so what you can see here are these results of these experiments. And what you can see is that, very briefly, I'm glossing over this rather quickly, that the cells that are predicted to be exhausted stay exhausted, and that the cells that are predicted to be memory-like, they stay with a large fraction stay memory-like, but also many of them become exhausted, which might make sense because these memory-like cells are reported and known to be a replenishing pool of new CD8 T cells that then turn out to be exhausted in this chronic infection setting. And with this, I'm at the end. So very briefly, as a summary, I talked about comparative single-cell data analysis, extending CellCnn to iCellCnn in order to classify on the basis of imaging flow cytometry data. And I showed you this demonstration to diagnose Sézary syndrome on single-cell morphology. I showed you how to do spatial single-cell analysis using this approach that we now, the current acronym is S to the 3 SEMA. So that's a supervised spatial single-cell image analysis. So there I showed you how you can use this approach in order to describe the tumor-specific cell type composition of these mate cells. And then in the end, I glossed quickly over how to do time series analysis using single-cell RNA sequencing, taking advantage of these velocity analyses to come up with simulation-based trajectories. And I showed you how to learn trajectories of T-cell exhaustion in chronic LCMV infection. And with this, I thank the group here in Tübingen and people that were involved in all of this work and funding, obviously. Thanks a lot. Looking forward to your questions.
Okay, many thanks, Manfred. That was really very interesting and definitely very relevant for Scailyte. So I really enjoyed the talk. Unfortunately, now there's a lot of questions popping up, and that means that I'm going to have to put my questions back. So I'll bother you another time. And I would actually start with questions from externals because they don't have as good of an access to you as people from Scailyte. So I would start with Imran Aslam's question, who asked, like, have you or Scailyte investigated when disease phenotype is not clearly defined, for example, as in multiple sclerosis or other heterogeneous diseases? In other words, can you discover data-defined phenotypes? I mean, I can answer that from a Scailyte perspective. No, we haven't. Normally, we look at data where the phenotype is defined, and we have some pathologist or some medical person evaluating and giving a clear label. So what about you?
Yeah, so we found that it's already sufficiently difficult to to come up with good models if the disease phenotype is well defined. And actually, I mean, we have done some work in multiple sclerosis where we did these types of analysis. So where we had flow cytometry data and we associated with disease phenotype. And so, I mean, we had clinicians that provided us with, for us, clear disease phenotypes, like patients suffering from the disease or not. Or another project that very recently was published after a marathon of actually getting this study published, where we investigated which cell subsets characteristically change when you treat a patient. So there we consider treatment of multiple sclerosis using dimethyl fumarate, and there we could identify a cell subset that we already, like before in a different study, identified as distinguishing healthy from diseased patients. But again, I mean, in order to get back to your question, this diffuse disease phenotype setting we did not consider. I think because it is difficult. Because I mean, if you have a noisy label for your supervised analysis, you somehow have to deal with it. Maybe in the end, then it boils down to do unsupervised analysis, where you just go ahead and cluster your single cell data and then try to see whether these clusters are somewhat associated with your disease, with your diffuse phenotype. I hope this answers the question.
Okay, thanks. Sounds great. Then there's a question from Gonzalo Rosso about the second part of your talk. So the question is, since the tissue processing and staining may vary even within healthy samples, and therefore that produces a broad range of fluorescence intensities, how robust is your MATE approach when accounting or correcting for different fluorescent intensities to differentiate healthy and disease?
I mean, the data that we considered, this codex data, was obviously pre-processed and normalized as good as you can. What we have seen is that this codex data is actually quite noisy, and we assume that this is rather related to artifacts of the segmentation that makes it quite difficult to assign cell types to the data that comes out of these codex measurements. Much more difficult than using flow cytometry data, for instance. I mean, one reason why we are actually not so concerned about this noisiness, this patient-to-patient variability in this data, is related to the fact that the analysis that we do is a supervised analysis, where we have a patient cohort of 30 patients, say, and then 15 of these patients are label 1, and the other 15 patients are label number 2. And then if you have this patient-to-patient variability that is unrelated to these labels, then our classification approach will simply work worse, but not produce false positive results. As long as these biases that these technical artifacts introduce distribute randomly across the patient cohort. And in the very particular case of the example that I showed you here, we actually had, for patients, we had biopsies, tissue slides that comprised both parts of the healthy tissue of the very same patient and the tumor tissue. So we don't suffer from systematic biases across technical biases across tumor and healthy tissue, because we have this matching within a single patient in this data.
Okay, very nice. Like a question from Martin from Articum from within Scailyte actually matches this topic, so I'm going to put that ahead of another question. So he asked like that some pathology slides seem to have multiple annotations, and how are these handled by your models?
Okay. I mean, so that was actually a strength of the study, that the pathologists went through the histology images and told us this part of the image is Gleason 3 and that part is Gleason 4 within the same image. And the way how the model deals with this is, I mean, is by not using the whole image as input to the model, but only using image patches from the model. And so the patches, like the subsets of the image that we provide the model as training input, are always pure with respect to one of the different labels. So, I mean, we don't make the life hard for the model and choose exclusively image patches that are at the border of these segmentations.
Okay. And what do you do later when you make predictions? Like, so say you want to predict whether a certain tissue is a certain Gleason score. Do you also just present homogeneous tissue, or would you present border, like tissue at the border?
So what you can do with the model is to go at every single position of the image, and then basically extract the patch around this position, and then let the model make a guess what kind of Gleason score you have. And then you get a map of possibly varying Gleason scores for this image. And then what in this Gleason scoring is done by the pathologists is to assess which are the dominant Gleason patterns in the image or in the tissue. And then the pathologists report the two predominant Gleason patterns. And that's the same thing that we can then do. We can then just say, okay, so how many pixels on the image are Gleason X, and or like which Gleason pattern is the most dominant with respect to number of pixels being assigned to this Gleason score in the image, and which is the second most frequent. And then we report just these two most frequent Gleason patterns. And this is actually what I showed in these confusion matrices. You might have noticed that the Gleason scores were not 1 to 5 on these confusion matrices, but they were, what was that, 6 to 10 or something. Which are this is what the pathologists report. They report the sum of the two most frequent Gleason patterns.
Okay, excellent. Very interesting. So time's pretty much up. One very quick question at the end from Sarah Carr, also from within Scailyte. For the Gleason scores, I mean, if you have 5 different Gleason scores, do you use them as independent categories in the model, or is it rather a sequential variable or something?
So we didn't model it as sequential variable. We just trained a multi-class classifier. And
okay, interesting.
Maybe you could do better if you do some ordinal regression or something. But we didn't try this out. I'm sure that somebody tried it out and maybe had an incremental improvement of 0.1% performance or something.
Okay, excellent. Okay, I think time's up, and I would like to thank you very much in the name of Scailyte and everyone listening here. It was a very interesting talk, and I think enlightening discussion as well. So thanks a lot, and bye-bye.
Thanks for the invitation.